Hello and welcome friends, myself Dr. Smiley Pruthi. I am national level faculty teaching biochemistry. In this video, I am going to take few questions which can help you revise some of your topics of biochemistry which are frequently asked, and this will help you in your upcoming exam, that is January 2018 NEET exam for your MD entrance. So let's get started. The first question which I am going to take is, Kinks in the protein structure are formed by which amino acid? Four options are given: glycine, lysine, methionine and glutamate. The answer to this question is glycine. Glycine, because of its flexibility, it is introducing kinks or bends in the structure, or we say glycine is mainly found in beta turns. So turns or bends or kinks, these are formed by glycine. Also one more amino acid, proline, will form the kinks. Okay, if both of these are given and they ask you kinks, then proline will be the best answer to be marked and the second best will be glycine. Okay. Next question is, a patient's lipid profile is given. They are giving you the levels of triglycerides, cholesterol and HDL, and the units are milligram per deciliter, and they are asking you LDL. How much is the LDL of this patient? So how to calculate LDL? We have FRIEDWALD'S equation, which is total cholesterol is equal to LDL plus VLDL plus HDL. So when we are measuring the lipid profile, then we are not measuring VLDL, we are measuring triglycerides. So total cholesterol is equal to HDL plus LDL plus VLDL is equal to triglyceride by 5. So in that case, our LDL, if we have to calculate LDL, then LDL will be total cholesterol minus HDL minus triglyceride by 5. This equation you have to use when the units are given in milligram per deciliter, when the units are given in milligrams per deciliter. But if the units are given in millimoles per liter, then almost the same equation, but now you will write triglyceride divided by 2.2. So now the answer will be millimoles per liter, right? So in this question, we have to apply this equation. LDL is equal to total cholesterol minus HDL minus TG by 5. So in this question, they are giving you these values. Total cholesterol is 300, HDL is 40 and triglyceride is 200. So the answer will be 220 milligram per deciliter, right? Next question is, lipoprotein lipase is activated by which protein here? ApoC1, C2, ApoC3, ApoA1. So answer is ApoC2. What is enzyme lipoprotein lipase? This enzyme lipoprotein lipase is an enzyme present in the endothelium of blood vessels. This enzyme breaks down the triglyceride into 3 fatty acid and glycerol. This triglyceride is present in chylomicron and VLDL. As chylomicrons and VLDL are lipoproteins which are moving in circulation as they are forms of lipid transport, so this enzyme which will break the triglyceride of chylomicron and VLDL, this enzyme is also to be present in blood, right? Now, the activator of this enzyme is ApoC2. Let's see the role of apoproteins. Various apoproteins have various roles in the structure of a lipoprotein. What do you mean by lipoprotein? Lipoprotein means lipid plus protein, and how it is arranged, the lipid portion which is non-polar, that cannot freely circulate in blood because blood is polar and lipids are non-polar. So for making them suitable to be transported in the polar blood, what body will do? Body will synthesize a periphery made up of protein. Protein is polar, you can say hydrophilic. So hydrophilic periphery and hydrophobic core. This kind of structure known as lipoprotein, example HDL, LDL, VLDL. So they transport lipids from one place of body to another place, right? Now we have proteins in this lipoprotein. Proteins are known as apoproteins. For example, ApoB48, ApoC, ApoA, ApoE, all these are proteins. They have various roles. So let's get started with the roles. First is structural role. As we need something in the periphery, so that's why that is known as structural role. And that structural role is played by ApoB. ApoB48 is in kilomicrons. ApoB100 is in VLDL and LDL. Next role is they can act as enzyme cofactors. Either they can be activator of enzyme or they can be inhibitor of enzyme. For example, ApoC2 is activator of lipoprotein lipase. We have just done that question. ApoC3 will inhibit the same enzyme lipoprotein lipase. ApoA1 will activate LCAT and ApoA2 will inhibit LCAT. What is this LCAT? The full form is lecithin cholesterol acyl transferase. These activators, these enzyme cofactors, this knowledge I have taken from latest edition of literature, that is 7th edition, which was just updated a month back. So I have taken it from latest edition, right? Next thing is that these apoproteins can also act as ligands for the receptors. Example, ApoE is the ligand for remnants, that is chylomicron remnant, VLDL remnant, or IDL, that is intermediate density lipoprotein. Then ApoB100 and ApoE, both are ligands for LDL receptors, but main ligand is ApoB100 and minor ligand is ApoE here. And also ApoA1 is the ligand for HDL receptor, right? Next question, tomcat urine smell is seen in. Answer is multiple carboxylase defect. So you can see few common urine odors which are asked. Example, with this one we have done. Next, mousy or musty odor, that is seen in phenylketonuria, which is defect in phenylalanine hydroxylase enzyme. Burnt sugar-like odor, that is maple syrup urine disease, which is defect in the catabolism of branch chain amino acid, and the enzyme deficient is alpha keto acid dehydrogenase. Next is swimming pool-like odor in Hawkinsinuria. Next, question number 5. Odd chain fatty acid can form glucose by which pathway? Answer is propionyl-CoA. Various other options were glycerol, acetyl-CoA entering TCA, lactate. This was a recently asked question. So let's start with basics. The basic is that fats can never be converted to carbohydrates. If they ask you the reason is irreversible link reaction. Link reaction is a reaction, one-step reaction between glycolysis and Krebs cycle, and this reaction is pyruvate gets converted to acetyl-CoA. If this is irreversible, that means pyruvate can form acetyl-CoA, but acetyl-CoA can never form pyruvate. Can I say pyruvate is mainly coming from carbohydrates, that is glucose, and acetyl-CoA is mainly forming fats in the body? So carbohydrates, excess carbohydrates can be converted to fats, but the opposite is never true, that is fats can never be converted to carbohydrates. This is very important. But now we have two exceptions also to this general rule. Two exceptions are glycerol and propionic acid. See, glycerol is a breakdown product of triglycerides and propionic acid is a breakdown product of odd chain fatty acids. So both these things are coming from fats, right? But they can be converted to glucose, that is carbohydrate. So these are two exceptions, right? So propionic acid or propionyl-CoA is glucogenic, right? But how it is glucogenic? Because it is forming, this propionyl-CoA is forming an intermediate of TCA cycle, that is succinyl-CoA. Any intermediate of TCA cycle can form oxaloacetate. OAA stands for oxaloacetate. So any intermediate of TCA cycle can form oxaloacetate, and oxaloacetate after so many steps can form glucose, right? So this way any intermediate of TCA cycle is glucogenic, this way propionyl-CoA is glucogenic. Let's see the detail of these reactions, how propionyl-CoA is getting converted to succinyl-CoA. Propionyl-CoA is a 3-carbon compound getting converted to dimethyl malonyl-CoA. Malonyl or malonate is a 3-carbon compound. If they say malate, Malate is a 4-carbon compound. Remember, this malate is intermediate of TCA cycle. Malonate, 3-carbon, is inhibitor of TCA cycle, right? So now here malonate or malonyl is written, that means 3-carbon, but full name is dimethyl malonyl CoA. So 1 carbon extra in the form of methyl. So this compound is 4-carbon. So now from 3 to 4 carbon, obviously we have to add carbon dioxide. So the name of enzyme will be propenyl CoA carboxylase. Any carboxylase uses biotin, that is vitamin B7. So this step also uses vitamin B7 or biotin. Next is dimethyl malonyl CoA gets converted to L-methyl malonyl CoA. So that is also 4-carbon compound, an isomer of this. So enzyme is racemase here. Capital D to capital L interconversion is always by racemase. Next is L-methyl malonyl CoA gets converted to succinyl CoA. Succinyl CoA is also a 4-carbon compound. Now again it is also isomer. Here intramolecular transfer is occurring. This occurs with the help of enzyme mutase, and this mutase requires vitamin B12, right? So this way propenyl CoA is glucogenic. One additional information I would like to tell you here. See here, B12 is required in this step. So in B12 deficiency, which acid will come in urine? L-methyl malonic acid will be coming in urine. In B7 deficiency, propionic acid will be coming in urine. These are also frequently asked questions. Next, I am taking 2 questions together because there is little confusion between these 2 questions. So I'll clear that here. Question number 6 is, muscles cannot maintain blood glucose levels because of deficiency of which enzyme? Question number 7 is, muscles cannot make use of glycogen because of deficiency of which enzyme? So these are 2 different questions. Be careful in reading the question. Before discussing them, let's see some basics. For the transport of glucose, we have GLUTs, that is GLU stand for glucose, T stand for transporters. These are glucose transporters which are mainly present in our body for glucose transport, and these GLUTs are bidirectional. Bidirectional means when glucose will enter a cell via GLUT, then it can come out also, but we don't want that. So for example, this is a cell, this is GLUT, this is glucose here. So glucose is entering the cell via GLUT, right? But the cell has to retain glucose. Cell cannot allow that glucose is going again out of the cell via GLUT. So this cell will do something here, and what is that? Immediately, quickly, the cell will convert glucose to glucose 6-phosphate, right? Because if it is glucose, then it can escape the cell via GLUT. But if it is glucose 6-phosphate, then it can't escape the cell via GLUT. So can I say that phosphorylation of glucose is done for the entrapment of glucose inside the cell, right? Okay, now let's see the next basic. Glycogen. Glycogen formation or breakdown in our body occurs in only 2 organs, that is liver and muscle, also to a minor extent in brain also, but mainly in liver and muscle. The function of liver glycogen is to maintain blood glucose levels. The function of muscle glycogen is for its own purpose, that is for muscle contraction, for muscle activity. So can I say that liver will give this glucose to blood, but muscle has to use this glucose or glycogen for its own purpose, right? So when glycogenolysis occurs in liver, then the glucose released should come out of liver cell into the blood, because liver has to maintain blood glucose levels. So the end product will be free glucose, because it has to come out, and it can come out via GLUT. But when muscle glycogenolysis will occur, then the glucose release should not come out of muscle cell, because muscle has to use it for its own purpose. That's why the end product is glucose 6-phosphate here, so that it cannot come out via GLUT, right? So therefore the enzyme glucose 6-phosphatase, this enzyme which will convert glucose 6-phosphate to glucose. This enzyme is absent in muscles and it is only present in liver, because we don't require this enzymes in muscles, because in muscles when glycogen is broken down, the end product is glucose 6-phosphate. After that, the next reaction, glucose 6-phosphate to glucose, has only to be occur in liver, right? So therefore, see question number 6. Muscles cannot maintain blood glucose levels because of deficiency of which enzyme? Glucose 6-phosphatase. So this is a very simple question, right? But glycogenolysis, that is breakdown of glycogen, during glycogenolysis, the first enzyme is glycogen phosphorylase, which is also the rate limiting enzyme. This glycogen phosphorylase releases 90% glucose in the form of glucose 1-phosphate, and the second enzyme is debranching enzyme, which releases just 10% of the glucose in the form of free glucose, right? So main enzyme is phosphorylase, which is doing this. Okay, so now see the next question. Muscle cannot make use of glycogen because of deficiency of which enzyme? Now answer will be glycogen phosphorylase, right? Because muscles cannot make use of glycogen if the very first rate limiting enzyme of glycogen breakdown is not there, then muscles cannot make use of glycogen, right? Normal situation is this, that muscles normally don't have glucose 6-phosphatase, but they can make use of their glycogen, na? So this is an abnormal situation, that muscles cannot make use of glycogen if this enzyme, the very first rate limiting enzyme of glycogen breakdown, is not there. Okay. Next question is Malate shuttle is important in. Options are glycogenesis, glycolysis, gluconeogenesis, glycogenolysis. This question is given in your guides also. All the books, almost all the books will give this question, but they'll give the answer as gluconeogenesis, but this is not the answer. Answer is glycolysis. I will tell you the reason also. See here, Malate shuttle. Let's first understand what is Malate shuttle. Basically, what is the purpose of Malate shuttle? Malate shuttle is for the transport of NADH from cytoplasm to mitochondria, because in mitochondria ETC occurs and this NADH has to enter into ETC for giving ATPs, right? And pathways like glycolysis, which are occurring in cytoplasm, they need that their NADH from cytoplasm should be transported to mitochondria, right? Can I say any pathway which occurs in cytoplasm that only requires shuttle? For example, link reaction and TCA cycle are also producing NADH. Do they require shuttle? No, they don't require shuttle because these pathways are already occurring in mitochondria. They don't require shuttle because they are already producing their NADH in mitochondria only, which can directly enter into ETC. There is no role of any shuttle here, right? So the next concept is about gluconeogenesis. I have to tell you. In gluconeogenesis, actually what happens? Gluconeogenesis starts from mitochondria, and the first step is pyruvate gets converted to oxaloacetate, and next all steps have to occur in cytoplasm. So this oxaloacetate has to come outside in cytoplasm so that this oxaloacetate after various steps can be converted to glucose. This is gluconeogenesis, synthesis of glucose from non-carbohydrate sources, right? But let me tell you, oxaloacetate cannot cross inner mitochondrial membrane. So in order to take oxaloacetate outside, we take the help of malate here. Oxaloacetate gets converted to malate. Malate can cross inner mitochondrial membrane, and then in cytoplasm, malate can again form oxaloacetate. So this way we take the help of malate here. But this is not malate shuttle because see the detail. Here NADH is used in mitochondria. Here NADH is produced in cytoplasm. So basically what is happening here? That NADH is taken from mitochondria to cytoplasm, which is opposite to malate shuttle, which is opposite to the purpose of malate shuttle. So this is not malate shuttle. Here our purpose is to bring oxaloacetate outside. We don't care what is happening to NADH. We just want oxaloacetate to come outside. So this is not Malate shuttle. Be careful. This answer is given wrong in your guides. So answer is glycolysis. Okay. Next question, number 9th. During prolonged starvation, rate of gluconeogenesis depends on options: increased alanine levels in liver, decreased cyclic GMP levels in liver, ADP in liver, or decreased essential fatty acids in liver. Answer is increased alanine levels in liver. Actually, see here, suppose this is muscle and this is fasting muscle. Here I am going to make liver and this is blood inside. So what happens in fasting muscle? Glucose gets converted to pyruvate and then pyruvate gets converted to alanine. This alanine then comes out of the muscle cell into blood, and this alanine is taken up by liver cells. Now in liver, this alanine will form glucose by which pathway? Gluconeogenesis, and then this glucose is supplied to fasting muscle. So this is a cycle made, and this is known as glucose alanine cycle, also known as Cahill cycle. CAHILL, Cahill cycle. You also must have heard of Cori's cycle. Cori's cycle is glucose lactate cycle, which is occurring in exercising muscles when anaerobic conditions are created. But this is Cahill cycle occurring in fasting muscles, which is done in case of fasting when alanine amino acid is used for gluconeogenesis, that is formation of glucose. Let me tell you, 18 amino acids out of 20, 18 amino acids can form glucose. And here you can see alanine is forming glucose. Let me tell you, alanine is the most glucogenic amino acid, right? The carbon skeleton of alanine, 3-carbon alanine, is used to make glucose. So obviously we'll be using 2 molecules of alanine to make 1 glucose, that is 6-carbon. So during prolonged starvation, rate of gluconeogenesis depends on increased alanine levels in liver, which are coming from the fasting muscles. Next is the last question which I am going to take in this video. Which of the following is a palindrome? Palindrome, you know, palindrome is sequence in opposite direction. And your guides give example of English word refer as example of palindrome. See, REFER from here, REFER from here. Is it palindrome? No. This is not a palindrome. You should never take example of English word. If you will do that refer as example of palindrome, then you will say this is the answer, TAAT. TAT from here, TAT from here, but no, this is not the answer. Answer is this. Let me tell you the basic point. Restriction endonuclease enzyme is cutting palindrome. But tell me, restriction endonuclease enzyme is cutting DNA or some English word? Obviously, it is cutting DNA, not some English word, and DNA is double-stranded, and those 2 strands are anti-parallel. No English word has such characters, right? So we can never take the example of English word. We always have to take the example of DNA only. Okay, let's first take this TAAT. If TAAT written like this, then obviously left is 5', right is 3' end, and the complementary strand will be 3' here and 5' here, and the complementary sequence will be ATTA. So read the upper strand. Let me tell you, whenever you read any strand, DNA or RNA, you always read in 5 to 3' direction. So if you read the upper strand, it is TAAT. Now read the lower strand, ATTA. This is not palindrome. They are not same, na? It is not palindrome. Answer is this, GGCC. If GGCC written like this means 5' here, 3' here, the opposite complementary strand will be 3' here, 5' here, and the sequence is this. Now read the upper strand, GGCC. Read the lower strand, GGCC. This is same and this is palindrome. So never take the example of any English word here. Okay. This is it with this video. Thanks for watching. Your best wishes to your future exams.